Well, hello and welcome to the second presentation of the 2021 Fish Tales Lecture Series, presenting the science of Great Lakes fisheries. My name is Mark Holy, and I will be your moderator for tonight's lecture. The Fish Tales Lecture Series is a collaboration between myself, a retired fisheries biologist, and Kagan Haringa, the naturalist and program director for the Crossroads at Big Creek Learning Center and Nature Preserve. Kagan and I welcome your comments about the lecture series and your suggestions for future topics. To provide your feedback, please send an email to fishtaleslecture@gmail.com as shown on the screen. Tonight, we are again pleased to partner with the Door County Library to utilize their expertise and facilities to host online Zoom and Facebook Live presentations. And I want to acknowledge Laura Karakhan and Morgan Mann for their support and guidance with presenting this lecture. Thank you both very much. If you missed our February 18th lecture on the science of catching and releasing angler-caught bass by Dr. Stephen Cook, you missed a good one. But you can access that lecture by visiting the Door County Library calendar for February 18th and click on the video link, or send an email to fishtaleslecture@gmail.com and I will share the YouTube link with you. And don't forget our upcoming lectures. On March 25th, we'll have Using Science to Control Sea Lamprey in Lake Michigan, presented by Jessica Barber from the Fish and Wildlife Service in Marquette. And on April 8th, we'll have Great Lakes Bass Marking Program for Trout and Salmon, Better Fisheries Through Better Science, presented by Charles Bronte from the Fish and Wildlife Service in Green Bay. All presentations are on Thursday evenings at 7 o'clock Central Time. With the expansion of both walleye and lake whitefish populations in the Bay of Green Bay, how far and where these fish move have become important management issues to ensure their continued sustainability. Do fish interchangeably move between the east shore and west shore of Green Bay, or from the southern basin to the northern basin, including the Bays de Knox? To present the science around these questions, tonight, Dr. Dan Isermann will speak to us on Where Fish Wander in Green Bay, Tracking Movements of Walleye and Whitefish. Dr. Isermann serves as the unit leader of the United States Geological Survey's Wisconsin Cooperative Fishery Research Unit at the University of Wisconsin, Stevens Point. The Cooperative Research Unit's program was established by the Department of Interior to enhance graduate education in fisheries and wildlife sciences, and to facilitate research and technical assistance between natural resource agencies and universities on topics of mutual concern. As one of only 40 such units in the United States, the Wisconsin Fishery Research Unit is a unique partnership among the Wisconsin Department of Natural Resources, the UW Stevens Point, and the U.S. Geological Survey. Dr. Isermann received his bachelor's degree from Southern Illinois University, his master's degree from Tennessee Tech University, and his PhD from South Dakota State University. Dr. Isermann conducts research on a variety of fish populations across the state of Wisconsin and trains graduate and undergraduate students who are eventually employed as fish biologists with state and federal agencies. Before coming to UWSP, Dr. Isermann also worked as a fisheries biologist for both the Ohio Division of Wildlife and the Minnesota Department of Natural Resources. We are all very much looking forward to your presentation. Dr. Isermann.
So thanks everybody for tuning in tonight. And first thing I want to do is recognize all the other folks here listed after my name. This is a really large collaborative research project involving lots of individuals. Specifically, we worked very closely with the Wisconsin and Michigan Departments of Natural Resources to address questions important to fisheries management through this process. I'd also like to recognize that most of the funding for the work we've done that I'm talking about tonight came from the Great Lakes Fish and Wildlife Restoration Act, which is administered through the U.S. Fish and Wildlife Service, and also from the Fox River Green Bay Natural Resources Trustee Council, And that funding is associated with the natural resource damage assessment funding available for that portion of Green Bay. And then a variety of other partners that helped make this happen. So as Mark mentioned, I appreciate that thorough introduction. I am the unit leader at the co-op unit here at UW Stevens Point. I work for the U.S. Geological Survey. Some of you may be not familiar with our cooperative research unit program. Mark highlighted it pretty well. It's sort of a threefold mission. A big part of that is conducting applied research to help our partners, which include state agencies, the Fish and Wildlife Service, and also various tribal entities. We also provide a lot of technical assistance to those entities. And then, as Mark mentioned, one of our big goals is sort of to train the next generation of fish and wildlife biologists. So there's quite a few things I want to touch on before we actually get to the fish. And one of those is I want to talk a little bit about the technology that we are using to answer these questions. It's a little bit more sophisticated than this technology depicted here. This is actually at a gas station in Brown County, and I took a picture of it because they are so rare these days. I'm not sure if it had a dial tone. I don't think I lifted it up, but we're using some things a little bit more advanced than this. A lot of you are probably familiar with radio telemetry. So this is where we're attaching collars to animals or putting tags in fish to track their movements. And sort of in a simple definition of this, each one of these tags or collars is on an individual frequency. So it's kind of like tuning in your radio dial in your car where you're seeking out individual stations. And that's going to be a little bit different than what we're going to do with the telemetry we're working with. And so we're using acoustic telemetry. And how this differs slightly is that each one of the tags is actually on the same frequency. So we're listening to the same radio station. They're individually coded. Yeah, somebody's not muted here.
Hello, could you please mute your computer?
So these tags emit a signal coded for each individual fish, and then we use these receivers, the black cylinders here to at least my right, that listen for these fish. And so we position these receivers at various locations in a variety of different ways. And when a fish swims by, it registers that tag with a date and a timestamp. And so the tags are fairly expensive and the receivers are fairly expensive. But I always like to put this in the context of the fact that we're working with fisheries that are worth at least tens of millions of dollars and potentially more. And so this is just a schematic of where we have these receivers located for the studies that we're doing now. In total, it's about 192 receivers. A lot of them you can see are in lines, and it looks like on the map that they're stacked right on top of each other, but they're actually about 1.6 miles apart. So there's quite a bit of room in between them for fish to swim through, but we've positioned them based on how well we can hear the individual tags. And then we also have receivers in many of the tributaries to Green Bay. We're not showing all of the receivers there. Every tributary has at least two, but some like the Menominee and the Fox River have several. And then one thing, for those of you that aren't that familiar with Green Bay, I want to point out Chambers Island here, where the white arrow is. It's going to be an important point or line of demarcation that we're going to talk about quite a bit. But you can see these lines let us know when fish might come out into Lake Michigan or go into a certain bay or tributary. And so in order to get the information off the receivers, we have to lift them up off the bottom, and then we download the data using Bluetooth technology. And you might ask, well, how is it possible to pull off a telemetry study of this size? And the Great Lakes Acoustic Telemetry Observation System is a big part of this process. And so this is a collaborative group of researchers from across the basin. The group provides both infrastructure, data analysis assistance, and has a really nice online data interface that allows researchers to upload their information so it can be shared. And the funding for this has come through both the Great Lakes Fishery Commission and the Great Lakes Restoration Initiative. And so this organization or this group has been really critical to us doing the studies that I'm going to talk about tonight. And then again, for those of you that maybe aren't that familiar with Green Bay, for many folks around the Great Lakes, you might think of it as not that big of a body of water, but it is the world's largest freshwater estuary, covering more than 1,600 square miles. And one of the important things to keep in mind is there's some pretty significant gradients in the bay. If we're down here near Green Bay in the south, it's relatively shallow, warmer, more fertile. A lot of that has to do with the inputs from the Fox River. And then as we move north into Michigan towards the Bays de Noc, it's deeper, more oligotrophic, less fertility, clearer water. And those gradients are going to be pretty important to some of the results that we're going to talk about this evening. All right, so now let's talk about the fish, which is probably why most of you are here. We're going to start by focusing in on walleye. The Green Bay walleye fishery is probably one of the best known fisheries in North America. It's a destination fishery. People travel from all across the continent to come to Green Bay to fish for walleye, specifically for really large fish. And so because of this importance, the species is also really important to both the Wisconsin and Michigan Departments of Natural Resource that work to manage this fishery. And so a few thoughts on the conceptual model that we had for Green Bay walleye. And a conceptual model is just merely how we think things work. And a few important components of this is that we think that walleye in northern and southern Green Bay are separate stocks, and that the stock in southern Green Bay is primarily supported by fish spawning in different tributaries, including the Fox, Menominee, Oconto, and Peshtigo rivers. And then in northern Green Bay, the assumption is sort of some of the population is coming into rivers to spawn, like the Whitefish in the Escanaba River, but that there's also spawning occurring in the Bays de Noc that helps support the fishery. And so this conceptual model we've formed has been based on a lot of previous work with conventional tagging. And so that includes floy tags and jaw tags that are attached to fish and then are recovered later by fishers across the bay. And then we get some information on where those fish have moved to, but it provides really limited information because we only know where the fish was captured. We don't know what it might have been doing before that. And then the distribution of fishing effort is not equal across the bay. So it could be that some fish go to places where maybe they have a low likelihood of being captured. And then we also base a lot of these assumptions on genetics work that's been done. And so this snippet at the bottom is just from a previous paper where we used genetics to show that the walleye spawning in those 4 tributaries in southern Green Bay appear to be the same stock. They're not genetically distinct. So one of the reasons that we started to look into walleyes in Green Bay, looking for the opportunity to maybe use telemetry to answer some questions, is we've seen opposite trends on both ends of the bay. So in northern Green Bay, the walleye population has declined. There's sort of a lack of natural reproduction. Some of that's probably related to a lack of zooplankton for young fish. And the Michigan DNR does use stocking to supplement this population. And some of the information from Michigan DNR jaw tagging has suggested that these fish are now being recovered further away from their tagging locations. And if we contrast that with southern Green Bay, where the population is really high, we have consistent recruitment. Obviously, it varies from year to year. That's a very different picture. There are some questions about walleye recruitment around Sturgeon Bay specifically, where historically there was a little more evidence that was occurring. And so Sturgeon Bay was a focal point of our tagging. So the objectives of our telemetry were to essentially test this conceptual model. And in southern Green Bay, we were really interested in, do most of the adult fish in that part of the bay spawn in these tributaries, or is spawning out in the bay also pretty important? And do some of these fish actually move north? And then the northern end of the bay, we were interested in where the fish actually go after they spawn, how many of those fish might be coming to southern Green Bay, and then whether or not they're all coming back to spawn in the northern part of the bay. So in order to do this, we obtained walleyes using a variety of different methods. We tried to get them from all over the bay. A lot of the fish came from nighttime electro fishing conducted by Wisconsin DNR. We also tagged a pretty large portion of fish during Michigan DNR's egg take up in Little Bay de Noc. And then we moved the fish to net pens or aerated tanks to make sure they were in good condition before we put a $350 transmitter in them and sent them on their way. We took a dorsal spine off each fish to estimate the age of the fish. And then each fish was fitted with a loop tag pictured here, denoting a $100 reward for the fish. And the reason we did this is if we got the tag back, we could reuse it. And we also had a temp logger on each tag. And in order to get the data from that temperature logger, we needed to recover it. And so these are the actual transmitters that went into the walleye. The black portion here is the transmitter that would register on a receiver. They had a 4-year battery life, and we sort of controlled this. These tags can actually last 10 years. But when you start to get too many tags out in the system, you can have some issues with tag collisions, and it could impede other research. And we knew after 4 years that would be enough to answer the questions that we had. So these tags will actually expire after the spawning window next year. And then this yellow portion that looks kind of like a rubbery dip here, that is the temp logger that I was talking about earlier. So to immobilize the fish during surgery, we actually used Electro immobilization. And so we accomplished this using a TENS unit, and it's pictured up in the corner of the screen here. This is essentially the same unit your doctor might prescribe to you if you had muscle spasms or muscle pain. You'd attach this and emit a low dose of electrical current. And this essentially sedated the fish, for lack of a better term, so we could conduct our surgeries. This is kind of a broad overview of our setup. So the entire time the fish is on the table, we're pumping fresh water over its gills. And then you can see the leads from that TENS unit. Those pads are tucked down along the fish, and it's secured in this foam surgery board. So it's padded and it can't really move around very much during the process. And then we close the incisions with a series of sutures. Then the fish went back in the tanks and we monitor them to make sure that they were in good condition before we release them. Usually, right where we caught them or very near to where they were captured. And so this became a bit of a traveling roadshow. Dan Demkowski and I traveled around Green Bay to a whole bunch of different locations. Essentially, if we could get our hands on some walleye, we would make an attempt to get there and put the transmitters into the fish. And so this is just a broad overlay of our initial tagging operations. At the end of our process, we ended up tagging 362 different walleye spread throughout the bay, including some fish in the actual Fox and Menominee Rivers, Sturgeon Bay area, and then some fish up in Little Bay de Noc in Michigan. The white circles denote fish where we only tagged in the spring in those locations. So a couple of the tributaries along the Michigan shore there, the Cedar and the Escanaba River. And so to kind of summarize again, it was 362 individual fish, a pretty good split between male and females. Fish were between 18 and nearly 30 inches, and their ages ranged anywhere from 2 to 22 years of age. And 63 of these fish eventually came, or the tags eventually came back to us. So we were actually able to reuse those tags and put them into new fish. So I just want to remind you of where the receivers are located. This is important because a lot of people, I think, have the impression that we have pinpoint locations of these fish all the time. And the only way we can detect them is if they encounter one of these receiver lines or installments, or they go into a tributary. And so it's just sort of a reminder. And again, remember, Chambers Island here is going to be an important line of demarcation for us. All right, again, to get the information, we got to go out and pull these things up off the bottom and download it. And when we go out, it's always beautiful weather. That's not true, obviously. Sometimes it's your fingers are pretty numb when you're punching the keys on the laptop, but it results in millions and millions of detections that we've got to work through. And so we're always in that process of sifting through the data to try to decipher what it means. And right now, I would call this a progress report. We have downloaded the receivers 3 times. We did miss a few this year due to COVID, but it's not going to result in a tremendous loss of information. The current tags for whitefish expired in November. And then again, I mentioned that the tags for walleyes expire this coming spring. So we'll download one more time in 2021 for these current projects. So again, this is just sort of a preliminary look at what we've learned. And so sometimes the fish aren't very interesting. And so this is a fish that we tagged in the Fox River, denoted by sort of that pink circle, and we detected it in the Fox River, but after that, we don't have any detections for it. And this fish was actually alive, but it didn't cross any other receiver lines. So all we know about it is that it stayed in southern Green Bay. And similarly, here's a fish that we tagged in Little Bay de Noc, where we detected it in Little Bay de Noc, but it never crossed another receiver line. So as far as we know, it's still in Little Bay de Noc somewhere, at least at the time that we pulled this data. But some of the fish are a lot more interesting. And so I'm going to show you a couple of these, and I'm not trying to suggest they're the norm, but they have a longer detection history. And so this is a female walleye that we actually tagged out on the lake side of the Door Peninsula. We were really curious if these fish came into the bay at various points in time. And I think this is an actual picture of the fish. I found it on my phone from that day. So it was a female. And we first detected it about a month later, just a little bit farther up the shoreline. And then in that same month, it had made it up near Washington Island. And then it moved over to Little Bay de Noc. And then it came down to Chambers Island in the same month. So this fish is moving around quite a bit in a short amount of time into Sturgeon Bay. And so at this point, we know that this fish was somewhere south of Chambers Island during the 2018 spawning period, but it wasn't in a river. And so that's sort of the scale of the info that we have based on our receiver deployments. Well, then that fish eventually showed up in the Fox River the following year during the spawning window, and we detected it all the way up in the Fox. It actually moved up into the river. So this year it looks like that fish is spawning in the Fox River. And then interestingly enough, the loop tag, that orange tag on the back of this fish was found on Cat Island. So it wasn't with the fish anymore. Steve Hogler with the Wisconsin DNR called us to say that it had been found on Cat Island, and we stopped getting information from that fish. So we don't necessarily know how it got there. We're assuming it died and maybe a bird got a hold of it, but that was sort of an interesting end to that fish's history. If we look at another fish, this is a much larger female that was 19 years old that we tagged up in Little Bay de Noc during Michigan DNR's egg take. And we first detected it sort of heading out of Little Bay de Noc in June and July. And then it was out towards Washington Island for a while. And so it wasn't being detected every day at this location. Just during that time period, that's where this fish was located. And then it comes out to the lake side of the Door Peninsula. And then it heads down to the Sturgeon Bay Shipping Canal, which is another way that fish can enter the bay. But it looks like this fish was out in Lake Michigan during the 2019 spawning window. And so we tagged it during spawning in 18 in Little Bay de Noc, but it's not back in Little Bay de Noc for the second year or the next spawn. Crosses into Sturgeon Bay, heads up to Chambers Island, heads back over to Washington Island, and then the last data that we have for the fish is back up towards Little Bay de Noc. And so again, during the 2019 spawning period, it looks like this fish was still out in Green Bay. It hadn't returned all the way to the back of Little Bay to Knock where we tagged it. And this fish very well still could be alive. We need another download of data to sort of add to its history. But if we kind of take a larger scale view of all of the fish collectively, which is the goal of the project, one of the big questions where do these walleyes that we tag south of Chambers Island actually spawn? And these circles represent the percentages. So down here, this 36%, that's the Fox River. So it's pretty important. A lot of adult fish that we tagged came down to the Fox, and that included fish that we may have tagged in the Menominee River in the fall. They eventually came out of the river and came down to the Fox. Same with Sturgeon Bay. Sturgeon Bay and the Menominee River also look to be fairly important. The Peshtigo and the Cano rivers are the low numbers. And what's really interesting is more than a quarter of those adults appear to spawn somewhere out in the bay, and they're not necessarily coming into a tributary to spawn. And so that was sort of the goal, or one of the primary goals of that exercise. So we also wanted to look at whether south fish are headed north and north fish are headed south. And so when we look at our southern Green Bay fish, only about 16% of those fish are crossing that Chambers Island line. And the other thing that's pretty interesting when we look at the data just coarsely is they don't spend a whole lot of time north of Chambers Island. It's almost like they push out into that part of the bay and turn around and come back relatively quickly. And very few of those fish enter the main lake, at least through that Washington Island gateway. If we turn around and look at the northern Green Bay fish, we see a different story. A pretty good portion of those fish come south of Chambers Island, and a lot of them spend at least a fair amount of time south of Chambers, and a pretty good number actually leave Green Bay and head out into the main lake. And so a little bit of a difference. Remember those gradients we talked about in terms of fertility and that water clarity and those sorts of things. So there's probably more to this, and we just need to dive into it a little bit more as we get more data. Folks are always really interested in how often an animal comes back to the same place to spawn every year. So we kind of pooled this information together. We actually had 111 fish where we kind of know where they were in 3 spawning periods. And remember, we have a 4th spawning period coming up here. 77% of these fish were detected in the same location or area during the spawning window in all 3 years. And while that may seem like fairly high site fidelity, compared to something like a salmon, it's relatively low. So there is a lot of straying going on. If we look at it in just 2 out of 3 years, 93% of the fish were in the same location in at least 2 out of 3 years. And what we don't know is if this represents some fish are actually skipping spawning in some years, or if they truly are spawning in different locations. We can't verify that an individual fish deposited its eggs in a certain location. So again, some of you are probably asking, why does any of this matter? So I threw a few things out here that seem relatively important. We showed that probably a fairly large fraction of those adult fish in southern Green Bay are spawning out in the bay. And this is somewhat important because the DNR indexes adult walleye using electro fishing surveys in those tributaries during the spring. And so we might be missing a portion of that adult population. And in order to account for that, we'd have to develop a different sampling strategy and possibly combine sampling strategies to account for the full population. And then what's really interesting and what I think is going to lead to some more telemetry research is this disproportional north-south exchange. These fish might be fairly well separated for spawning, but again, we're seeing these northern fish come south and spending a fair amount of time south of Chambers Island, and more so than was indicated by the previous tagging efforts. And this has implications potentially for other species. Some of our other research has shown that the walleye population in Green Bay could affect other species like yellow perch. And so now we have potentially these other fish moving south that might be influencing things that are going on in southern Green Bay. All right, so we're going to kind of abruptly transition to whitefish. I couldn't come up with a good segue today. Many of you are familiar with this fish and are aware that it supports critically important commercial and tribal fisheries around not only Lake Michigan and Green Bay, but around the Great Lakes. It's probably our most prominent commercial species that we have in the basin. And then more recently, it's really contributed to a very popular recreational fishery in portions of southern Green Bay. This has also become a destination fishery where folks are traveling from around the continent to come fish for these fish on the ice. And so that's sort of a new twist on managing this species. We have a new source of harvest besides our longstanding commercial and tribal harvests. And so maybe some of you aren't familiar with exactly how whitefish harvest is managed, but this is an interjurisdictional fishery. Obviously, even just around Lake Michigan, we have multiple states that have lake whitefish harvest occurring within their management zones. And again, we have these different components: commercial, tribal, and in some cases, recreational fishing. And so we use these geographic management zones that are depicted on the map. And how we determine safe harvest is based on these statistical models where we feed in our survey data and they provide estimates of how many fish essentially are available for harvest. And then we base our safe harvest numbers on those model estimates. And so sometimes these are calculated for individual zones. Currently and in the past, Wisconsin has used a single model for their 3 management zones that I'm kind of hovering over here. But what really complicates all of this is we have multiple stocks of whitefish in Lake Michigan that are producing fish. And these circles or squares aren't entirely accurate. I just want to give folks get it in their heads that there's these different groups of fish that are contributing to these fisheries. And these fish don't know where these lines are. So they're moving across these boundaries in some cases. And this presents a problem for trying to manage harvest and work with individual stocks. So we're going to really zoom in here on Northwest Lake Michigan and Green Bay specifically. Again, one of these conceptual models that's probably changed fairly recently, but this is kind of how we thought about things. Out in Big Bay to Knock, we had a group of whitefish that were spawning up there that were considered their own stock. Out here on the lake side of the Door Peninsula, and I'm just using North and Moonlight Bays as one point here because a lot of sampling has occurred there. We've got a group or a very large group of fish spawning on a lot of habitat out here. And we kind of viewed that as a different stock than the fish that were in Big Bay to Knock. And then for a long time, we didn't really put a lot of emphasis on what was going on in southern Green Bay. There wasn't a lot of evidence that natural recruitment was occurring at any great level. A lot of the previous work essentially assumed these fish were probably pretty similar to the fish that are out here on the lakeside of the Door Peninsula. But there's been a lot of changes over time. And one of the biggest changes has been the resurgence of tributary spawning in some of the tributaries to Green Bay, including the Fox, Econo, Peshtigo, and Menominee Rivers that we also talked about for walleye. So that's a pretty big change or difference in lake whitefish populations moving forward. Some other changes, following the tributary expansion, we've seen the population in southern Green Bay expand. We're pretty certain that's not solely due to tributary spawning, that there are fish spawning out in the bay. That winter recreational fishery has emerged, and now we're seeing harvests that do exceed 100,000 fish per year. And what's really interesting and has caused a lot of research interest, at the same time this expansion is going on, we're seeing declines in recruitment in other parts of the lake. And so that includes Big Bay to Knock and North and Moonlight Bay, where it appears that we're starting to see some signs that these populations are, at least recruitment wise, declining to some degree. So there's a lot of questions that we could answer with telemetry. And a lot of that is related to defining these stocks, not just with genetics, but also in terms of where are they and when. And we didn't have a lot of information on how these fish spawning in the tributaries moved around the basin. And so our telemetry work was really a lot about who is where and when are they there and how might they be exposed to different fisheries. And that's important to safe harvest allocation and also important to ongoing treaty negotiations that are happening for portions of Michigan waters of Green Bay. And so when we started this process, there wasn't a whole lot of information on telemetry with Lake Whitefish. And a lot of people consider them to be a relatively fragile fish, at least compared to walleye. So we did some surgery trials. These were conducted in Menominee Dam. Eagle Creek Renewable Energy was nice enough to let us use the facilities that we use for lake sturgeon passage to bring some whitefish in and actually go through the process. And so we did the surgeries, held them in these tanks for 48 hours. And this is a group of those fish just a couple hours after the surgery. Oh, for some reason that didn't play. And you can see they're swimming around pretty well. We got one fish back here that's not doing real well. That actually wasn't part of our surgeries, but they survived this process really well. And so then we took this out on the landscape and started tagging fish out in the lake. And so these were our primary stocks or tagging locations that we focused in on. Again, Big Bay to Knock, the lake side of the Door Peninsula, which we tagged around North and Moonlight Bays, and the Menominee and Fox Rivers. And we had a few tags left over, and we did tag a few fish in the Peshtigo River as well. How did we get these fish? Well, again, we used DNR electro fishing in the rivers, and then we had a variety of commercial and tribal fishers help us get our fish out in open water along the lake side of the Door Peninsula and Big Bay to Knock. And we really thank those folks for working with us to get those fish so we could do the study, because this is not something we can do with our fleet of vessels on our own. We held the fish in tanks again to make sure that they were in good shape before we put that expensive transmitter in them. And then they went through the same process as a walleye. The transmitter is quite a bit smaller and the battery life is only about 3 years. And the temp logger, as you can see, this yellow circular thing up in the picture wasn't attached to the tag, and that just made it easier to actually get the tag into the fish. And then at the same time we're doing this, the DNR and a bunch of commercial fishers were working to get these green loop tags into whitefish as well. And so we have sort of this secondary tagging going on, and they were able to tag a lot more fish than we were, but it kind of gives us two sources of information about fish movements. And we also took a fin clip for genetic purposes. And so I'm gonna show the preliminary results for these fish using these hotspot maps. And we're frantically working to update these because we have a lot of new information. I don't know that it's going to change the story any, but we are working to update them. And so the way these work, each circle is a receiver. We're not showing every receiver in every line or in every tributary, but the redder the color, the more fish that were detected at that location or in that line of receivers. And so an X means we didn't detect any fish, and white or really light colors mean very few fish. And so that's sort of the strategy that we're going to use to convey the info. So let's look at the Fox River. This black triangle down here denotes approximately where we tagged the fish. And then you see a bright red circle at the end of the river. And that's because we detected most of the fish, nearly all of them leaving. But then you don't see a whole lot of other color out on the landscape. You know, there's a few open white circles by Chambers Island and out here by Washington Island, but very few fish are making it to those receiver lines. And what this tells us is that these fish are staying south of Chambers Island. Again, Chambers Island is right here in the middle, and the loop tagging recoveries also reflect this. All right, let's move to the Menominee River. Again, the black triangle is where we tag the fish. That one red circle there is essentially those fish leaving the river. And then we see a little more color here. And what we saw is about a third of these fish did go north of Chambers Island. And some of them did leave Green Bay, very few, 6 of them at last tally. 2/3 of them, though, appeared to stay south of Chambers Island. So that's a pretty significant proportion. Now let's move out to the lake side of the Door around North and Moonlight Bay. 2 different tagging sites denoted by those black triangles. Again, a lot of red right around the tagging, but then not a lot of color. You know, a few of these fish are coming into Green Bay, but not very many. and not very many of those spent any time south of Chambers. So for the most part, as best we can tell at this point in our data, these fish were actually staying out in the lake. And so that's pretty important information for management. And again, the loop tags provided similar insights. So it wasn't just the telemetry. And then big bait and knock, the big bait and knock fish are a little more interesting. We tagged in a couple different locations, and you just see a lot more red and pink on the map. And so a good portion of these fish were actually leaving Green Bay and coming out into Lake Michigan. And some of these fish were also going south of Chambers Island. And so a little bit more mobile than those other tagging locations. And so to get back to why this is important, remember those management zones that I talked about at the beginning, where initially, or in the past and even currently, Wisconsin's using a single model for all three zones. And what our telemetry data and the loop tag data and our genetics information tells us is that it might be better to separate these out into two different groups. And this is sort of in a proposal stage. It's in the public meeting portion of trying to sort of decide whether this is a good approach. But based on our telemetry, remember, not very many of these fish out spawning on the lakeside are coming into the bay, and not a lot of these bay fish are coming out into the lake. And so there's some support for this approach using our telemetry information. It's not the sole source of information, obviously, but it's one way that the information is being used by fisheries managers and stakeholders. The big bait and knot group of fish has spawned more research interest because they're a bit more mobile. And so I've just kind of coarsely overlaid those management zones here. And even with our fairly simple telemetry array, you can see these fish are ending up in multiple management zones. And that makes estimating how many of these fish are out there and managing harvest a bit more complicated. And so we're planning to move forward with telemetry in and around Green Bay. Our next big focus is to do some more whitefish work. One of the things that we're going to do is change our array to a grid rather than those lines. And this will give us better more pinpoint information on where fish might be within the bay. What we're really interested in is for the commercial fisheries occurring in the bay and that recreational fishery, where do those fish spawn? How important is the Fox River? How important might spawning out in the bay be to those fisheries? And so our plan is to tag fish out in the fishery and then see where they go to spawn rather than tag them during the spawning window. So our plan is up to another 500 transmitters over the next couple years, and we're working on securing the funding to do that right now. I'm not going to talk much about this, but I mentioned the temperature loggers, and we are getting a lot of really interesting information from those loggers when they come back to us. This is just an example from one whitefish. It records the temperature every 4 hours. And so you can see during the winter time, there's probably not a lot of variability in water temperature to begin with. But when we get out into summer and fall, these fish are making pretty substantial movements across temperatures, even during an individual day. And we're not sure if that's, up in the water column and down in the water column or inshore or offshore. But as we piece this together, we'll start to get a better idea of what temperatures these fish actually inhabit during the time that they're out there. And then we've also used the technology for several other research endeavors. One of the more interesting ones has to do with lake sturgeon passage on the Menominee River. I'm going to show you this little video where we're using this fish elevator in the dam to capture sturgeon and then move them upstream above 2 dams to better habitat. And what we want to know is that the fish stay above the dams to spawn. And so we're using this same technology to answer that question. And we have shown that for the most part, they'll stay above the dams to spawn, which is the first step in assessing whether this passage program is successful. And then another application of it that we've used has been focused on muskies in Green Bay. This is another destination fishery for trophy fish where people travel across North America to come to Green Bay to chase large fish. And our interest here was we're not seeing a lot of signs of natural reproduction of muskies, and that's the goal of the restoration process, is to have a self-sustaining population. And so what we did with the telemetry is use it to guide us to spawning locations. And then we measured habitat attributes at those locations to try to determine what kind of bottlenecks may exist in these fish successfully hatching and producing offspring. So this was just another really useful application of our work. I don't know if you can see it, but there's actually a Floyd tag on one of those muskies, and it was one of our fish that we had a transmitter in. So we were able to sort of track it down to where this is probably a spawning event happening or getting ready to happen. So with that, I'm going to leave it there. I really thank everybody for tuning in, and I'm happy to take as many questions as we can field in the amount of time that we have left. My email is down here at the bottom of the slide, so feel free to reach out to me, and we can try to address any other questions folks might have.
Thanks, Dr. Isermann. A very interesting talk, really interesting stuff. And it's great to get this level of information to be able to make management decisions. So I'll just start taking some of the questions right from the chat that people put in. One of them asked, can the loops get caught on something and be a danger to the fish?
It is possible. We have had rare instances where people have actually snagged the loop with a lure. The reason we went with that tagging approach is because it has a much higher retention rate than a lot of the traditional tags that we were using, like a Floyd tag or a jaw tag. And so we really wanted to do our best to make sure that people knew there was a transmitter in the fish. And so it's kind of a trade-off. But we think the instances of fish getting caught or hung up by those loop tags is probably relatively small, but it can happen.
Okay, Benjamin asked, when will the $100 reward program expire?
I mean, I think we're going to keep it going as long as people keep sending us tags, because the temp loggers are good. You know, it doesn't matter when we get that logger back. You know, there's probably some kind of shelf life, but we'll continue to pay it out. And we're probably going to be tagging more fish into the future. So we're just going to continue to do it. We're not getting a whole lot of fish back anymore because, you know, natural mortality also removes some of those fish from the population. And, you know, you start with 300 and some, and over time, you just start to lose fish naturally. And one thing I would tell people is the loop tags do come out eventually. So some of the fish that have been out there for 3 years or 4 years almost might not have a loop tag. And so if they're cleaning fish, it doesn't hurt to look for the transmitter, because the transmitter is the part that gets you the $100. It isn't the loop tag. We want that black cylinder that's inside the fish.
Another question is, what is the effective or maximum range between the transmitter and the receiver?
So that can vary quite a bit in relation to noise, like if you're in a river versus open water. And the size of the tag. So obviously those walleye tags that are bigger, we can hear a lot further away. I think for the whitefish tags, roughly we would probably say a 1/4 mile or so as an average, and probably more like a mile for the walleye tag. So it is entirely possible that a fish could swim through a line and not be detected, but it would have to do it really quickly because the tags ping, you know, every minute and a half or so. And you know, they could somehow avoid, it's probably more unlikely to occur in a river where the fish might be around the receiver for a little bit longer period of time. And so one of the reasons we have more receivers in the rivers is because our tag detection can be lower. And so to overcome that, we just put a few more receivers out.
So kind of a related question for me is, how many receivers did you have across near Chambers Island?
Off the top of my head, I don't know the answer to that. I think like each of the half lines is probably more than 20. I mean, even looking at that that figure. I mean, roughly every mile and a half, but I don't, off the top of my head, I can't
Okay.
remember.
Well, but you had them every mile and a half.
Yeah.
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 Okay. So another question is there a website where the tagging and tracking information of the walleyes can be found?
So we're actually working on that. There is a website where you can find information about the study, and that's affiliated with Glados. We're about ready to launch some maps that have the muskie information on them, but we don't have like those histories I showed you guys tonight for those individual walleyes. I mean, there's 362 of them. And so we don't have those individually pieced together for every single fish. Eventually we will, and we'll distribute those in the same fashion. But I think we're probably going to use like our website and the Glados website as conduits for distributing that information. And we have people emailing us all the time about it, and we do intend to release that information. A lot of people, though, I think are surprised because they think we're going to know where the fish are all the time, and we just don't have that level of information. But we're working on it. It's really slow. I mean, I'm gonna guess that we're well over 10 million detections at this point in terms of data to sift through.
Okay, the next question is, of the 27% of walleyes that spawn in the lake, were those, or wait a minute, it says that spawn in the lake, were those fish spawning? I think what he means, or I think what the person means is in the bay, the 27% that didn't run up a river.
Yep.
Are they actually spawning in the bay?
And so, like I mentioned, we can't confirm that they deposited eggs out there. So it's very possible that they're not spawning. So that would mean they're skipping because they are mature. I think when we download our receivers again this year, that's going to provide us some really good information. So let's say we see a fish come into the Fox, and then the next year it's out in the bay, and then the next year it's back in the Fox. Like that would be at least a little bit more evidence that a fish might be skipping a spawn. But currently the technology doesn't allow us to verify that the fish has actually, deposited eggs, and it'd be really hard for a male to know that it had spawned. So we just assume they did, and we use where they were during the spawning window to make that call, but it does not guarantee they spawned out in the bay or in a river. I mean, they could have gone in the river too and not spawned.
So did you tag, or did you try to tag only adult fish?
Yeah, so you saw the range, like the smallest fish was just a little bit under 18 inches. They were all, should have been sexually mature and should have spawned in the next available window.
Okay. And then for the walleyes, this person said, my rough calculations reveal that if there were approximately 2 million detections, each walleye would have triggered 5,000 detections. And does that sound accurate?
Some fish trigger 5 times and some fish trigger 250,000 times. It depends on how much they contact the receivers. So you could imagine if they swim into the Menominee River and they sit within 100 yards of a receiver, we're going to detect that fish every minute and a half all day, every day until that fish leaves. And so that fish is going to have a lot of redundant detections. But a fish that's out in the bay and might just swim by one receiver or two, we might only have a couple of detections for that fish.
So it's a bit of a challenge to decipher what all that means then.
Yeah, I mean, the hard part is when you do get a fish that, you have hundreds of thousands and they're all pretty redundant, is just sort of synthesizing that into one piece of info. How long was the fish here? That sort of thing.
So the next question is, what new hypotheses have this current research spawned, or what's the next ones that you're going to address or try to address? I guess you got into that a little bit with your grid instead of a line, but
yeah. So for the whitefish, we're definitely interested in trying to identify where the fish that are out in the bay, where they actually go to spawn, like what percentage of the whitefish, say, ice anglers are catching, end up spawning in the Fox River versus the Menominee versus out in the bay. For walleyes, we're pretty interested in doing some more work in northern Green Bay where we're seeing issues with the population, and we're really curious whether movements might be related to the size of walleye, if larger females are moving farther distances in order to acquire enough energy to maybe spawn again the following year. So I think we'll probably be tagging more fish in that location. And you could always argue that we need to tag more fish in southern Green Bay to really answer our questions, and that's always an option as well.
So the next comment was, this was very interesting, and has this technology been used to track Asian carp?
It has. There are receivers positioned throughout the Mississippi River drainage that are doing this exact same thing with silver and bighead carp up into the Missouri River, into backwaters. And so folks with Fish and Wildlife Service, USGS, and a bunch of different universities are using that same technology to track individual fish.
Hopefully none of them have showed up in your grid though, right?
No, we get some really interesting stuff, and that's the beauty of that Glados network, is you have all these people sharing data. And so just last week, one of the muskies that we tagged did leave Green Bay. It ended up in the Muskegon River, and then it was recently detected in the Kewanee River. And most of the fish didn't do anything nearly that exciting, but these fish they can go quite a ways. I mean, and the only way you know about it is if you have this network that allows you to tap into all of those receivers. I probably should have shown a plot of all the receivers in the Great Lakes. It's pretty impressive at this point.
Yeah, and I was just going to mention there too, that the whole Glados organization or effort was started primarily by the Great Lakes Fishery Commission and was funded significantly by the Great Lakes Restoration Initiative.
Yeah,
that has been coming from the federal government. But there are receivers all throughout the Great Lakes. So they have programs like this just being done on every lake somewhere with the most important questions. So here's one, maybe I missed it, but were there many angler returns of whitefish, thinking and wondering about ice fishing mortality?
So the interesting thing is we didn't get very many, less than a handful from the anglers on the ice. Most of the returns came from our commercial folks. And I mean, that's not too surprising in a lot of cases, because we were actually tagging, right where these guys were providing us the fish. And so there may be something to that, but we were a little bit surprised. I mean, Scott Hansen would have to verify the returns from the loop tags, but we had very few whitefish from that winter fishery. And so that makes me think like, well, maybe that means a lot of the fish that are supporting that winter fishery aren't from these two rivers, and they're actually spawning out in the bay. But that's purely speculation. But that could be a reason that we didn't get a lot of our fish back, as we weren't necessarily focused in on the right spawning groups. Whereas in Big Bay to Knock and on the lake side of the Door Peninsula, where we were tagging fish essentially right out of the nets, we're probably more likely to recover those fish.
So a follow up on the carp question. Are you familiar at all with the results down there in terms of the migration or threat of fish getting into the lake?
I am not. And so there would be information on those fish accessing, like the Illinois River. There's receivers in the Illinois River. I'm not familiar with the results of those studies, but they are out there. I'm going to guess a fair amount of it is still in sort of the data analysis phase, but I would say, like, if I was going to Fish and Wildlife Service folks, I know people in the La Crosse office have been working on that telemetry and all up and down the basin. I mean, there's another good place to look for information would be the U.S. Geological Survey's office in Columbia, Missouri. They do a lot of work with silver and bighead carp if folks were looking for that info. But I don't, off the top of my head, don't have any information on that.
Well, that's the end of the comments that I received through the chat. Thank you once again, Dr. Isermann. That was a great presentation. If any of you have comments about this presentation or the previous one, please send them to fishtailslecture@wisc.edu. @gmail.com. Our next presentation is March 25th on sea lamprey control. Thank you very much. Have a good evening.
Yep, good to see all of you. Thanks for tuning in.